Okay, so since it is already 3 p.m., let's get started with the lecture. Once again, I just take my laser pointer. Yeah, so let's get started. So, welcome everyone to the last session of this last TA session for the course Bioengineering and Interface with Biology and Medicine. And the course code is NOC 25 BT05, and I am Arjun, and I am one of your TAs for this course. So we have already completed 8 weeks of this TA session for this course, but today I have asked for one more extra session for of TA course. So what we will be doing in this session is we'll be going through a general recap of the whole course, whatever concepts we learned in the whole course, and then we will be solving some sample problems which will be useful for your examinations. So we will get started right away. I don't think anyone is in the room right now, but anyways, I will be uploading this video in YouTube. So if you have any doubts after this, I'll also post the link for the uh, this YouTube recording as, and I'll also upload the slides in the Google Drive. So please feel free to go to the Google Drive Excel sheet, and then you can access the list of recordings. And if you have any doubts, you can also post that in the YouTube comment section. So I'll get started. So we covered basically a lot of concepts in this entire course, bioengineering, and how we started the course, how the course instructor, Dr. Sanjeeva Srivastava, started the course, is he tried to link the fields of biology and engineering. So bioengineering is a field which combines both these fields, and basically we saw how we will be able to bring together these two fields to solve problems. So first, in part one of this lecture, say part one of week one's lectures, so we saw why do we need to bridge the gap between biology and engineers, and for that the course instructor gave some problem statements which mostly revolved around feeding the world population and addressing the population growth problem, and he also addressed some other problems like the increase in environmental stress and calamities, and resulting in the problem generate increasing problems in with respect to food production. And he also laid after that he also gave some solutions as to how we can tackle this problem, and he also gave some examples and by how using DNA cloning and genetic engineering, how we can increase the plant production, and how we can generate crops which are resistant to certain disease pathogens or disease environments. And we also saw about a small set of molecular biology where we discussed about plasmids and water plasmids. So they are small circular extra chromosomal DNA segments, and we saw how plasmid can be engineered to insert our gene of interest, and and we also saw how certain restriction enzymes are used to cut the plasmid into pieces, and how the plasmid technology is used to make transgenic crops to provide more desired traits. And we also saw the common phages or vectors, plasmids used in the genetic engineering, mostly the bacteria M13 phage. And we also saw other problem statements like tackling climate change and global warming, and sorry, excuse me. And we also saw other problem statements like combating deadly diseases like HIV. And in addition to that, we also saw how to and we also saw the major challenge that we have to in today's world, which is cancer. And we also saw the general statistics regarding cancer, the incidences and mortality rate regarding cancer, and the different types of cancer, like majority of cancer like oral cancer, lung cancer, cervical cancer, and breast cancer. And we also saw another global problem that we are facing with respect to water, food, and nutritional energy. And we also saw that how we have only limited energy resources, and how we need to search for alternative energy sources. So, and in search of alternative energy sources, how we can go for um, how we can go for fuel cells, microalgae, cyanobacteria, as well as biofuel. Just give me a minute, I'll be back in 1 minute. Yeah, sorry. We saw all about those in the first part of the lecture, and in the second part of the lecture, we saw how bioengineering strategies provide solutions. We saw how biomimicry is a major part of the bioengineering process, and how the lotus leaf and the termite mold cooling and sonar technology from dolphins, as well as the kingfisher beak, which is also used in several real life engineered phenomena, and how there are mega projects such as Google X and Google Brain and supercomputers developed by IBM, which help in storage of large data and usage of large data to provide meaningful solutions. We also saw more about the revolution in medical research, where we saw about personalized omics technologies, and we also saw some of the biological based research which is going on in IIT Bombay. And after that, in week one, we saw more about life processes and cell, which is given here. So we started by discussing about what is life and what are the energy processes and evolutionary adaptation that living organisms undertake to survive, and what are all the qualities that a living organism need to possess in order to classify themselves as living, such as movement, respiration, growth, and reproduction. And after that, we also saw more about the organization of life, uh, ranging from information transmission, flow of energy and matter, and then evolution. And then we also saw how different organisms have distinct morphology with remarkable biochemical commonality. And then after that, we saw about the structure and cells, which are the structural and functional unit of life. And we saw about the two main types of cells, prokaryotic cells and eukaryotic cells, and the main differences between them. And we also saw that uh, the organization of life, ranging from a small atom which goes on into a molecule and results in a biosphere. And we also saw more about the evolution of my certain cell organelles within the cell itself. And we also saw more about the definition of cell organelles and what are all their general functions. We saw more about the Golgi bodies, peroxisomes, mitochondria, cytoplasm, and plasma membrane. And in the end, we also saw more about the cell communication, wherein we discussed about growth, division, and death. And then we saw more about cell signaling, where cell signaling is divided into four types of signaling, which out of them, which is called as autocrine signaling, paracrine signaling, endocrine signaling, as well as direct signaling. And in the towards the end of the week one's lectures, we saw about a clinician's perspective, where we saw about Dr. Aliasgar Moyedi, who was talking about glial cells, glioma localization of tumor, and removing it completely. So, following that, in week two of lectures, we saw more about the nucleic acids and central dogma. We saw that the central dogma is the conversion of DNA to protein, DNA converts into transcripts into RNA, and RNA translates into protein, and this is known as the central dogma. DNA is used for storage and transmission of genetic information, while RNA and protein is RNA. The conversion of RNA into protein is used for synthesis, and protein has several functions in our body. And after that, we saw the how the nucleic acids and nucleosides and nucleotides are formed, and we also saw how Watson and Crick came up with the 3D structure of the DNA, and we also saw how Linus Pauling's hypothesis and Franklin's X-ray crystallography data, as well as we discussed about the Chargaff rules for DNA. And after that, we saw how the genetic material, which is DNA, is compressed into chromosomes using histone modification, as well using wrapping on a histone octamer and compressing itself to form chromatin and chromosome. And then we saw more about the genetic gene regulation using Lac-Operon. And then we also saw a general timeline of discoveries involving DNA. And we also saw the different classes of RNA regarding which are notably mRNA, tRNA, and rRNA. And then we also saw the omics and personalized medicine, which is used to which is used and which is revolved around the usage of DNA and proteins. After that, we also saw more about the DNA tools which is used. Yeah, basic DNA tools which is used in biotechnology. We first saw about the DNA cloning, and where we saw already about the plasmid, which are extra chromosomal molecules and have few genes, where these plasmids can be used as a cloning vector, which has a DNA molecule that can carry foreign DNA into a host cell and replicate there. And then we saw more about the steps involved in DNA cloning, where the steps involved in cloning involves formation of recombinant DNA molecule, which is the rDNA, and then transformation and then selection. And we also saw how can we test for the fragment size using gel electrophoresis after the desired DNA is formed, and how do we interpret the gels from a vector map. And then we also saw a live demo of how can we do that. And in week 3 was a continuation of week 2, where we saw about more DNA tools which is used in biotechnology, where we started with discussing about the concept of PCR, which is polymerase chain reaction, and we saw about the steps involved in PCR, which is generally denaturation, annealing, and extension or elongation. And then we saw how the primer should be designed carefully to uh, not and primer should be designed carefully with to help introduce changes and study effect of those changes in the specific DNA. And then we saw our other ways by which we can other tools by which we can study DNA, which use mainly involve the use of 613 tags, and which involve the process called as a affinity chromatography. And after that, what happened is we also discussed about certain concepts which involve the use of PCR as well as this affinity chromatography, mainly in the use of production of insulin, human growth hormone 2, as well as this BCR-ABL gene enzyme system. And we also saw more about the transgenic codes for antithrombin production and other products which are generated using this DNA tools using likely tissue plasminogen activator and human growth hormone, etc. And then we saw more about gene sequencing through the Human Genome Project. And then we got an introduction into Sanger sequencing and how the basic process of Sanger sequencing happens, and how the Sanger sequencing also makes use of the DDNTPs, which are the dideoxynucleotide triphosphates, which helps in stopping the extension of the DNA and DNA extension. And we also saw more about the next generation sequencing technologies. And then we also saw about other technologies which are used to study which use DNA, such as in situ hybridization, reverse transcriptase PCR polymerase chain reaction, as well as DNA microarrays. Specifically, DNA microarrays will come in a lot of lectures in the following slides, as it is a very important tool which uses less sample volume and more samples can be analyzed in a single go. And we also saw the fundamental impact of food production through abiotic stresses, and how biotechnological solutions using this DNA tools help us in overcoming those problems. We saw more about genetically modified organisms and how we are able to develop stress tolerant plants. We also the example of that is golden rice, which have a increased carotenoid production, which is used to tolerate pathogens. And we also saw other examples like Bacillus thuringiensis, which is used to produce BT crops. So following that, in week four and week five mostly involved genetics, where we saw some basic definitions regarding the definition of gene and allele, and then we also got a definition of genotype and phenotype, as well as homozygous and heterozygous allele. And we also saw more about Mendelian genetics, how a scientist named Gregor Mendel performed experiments using pea plants to come at the Mendelian laws of genetics, which are mainly the law of segregation, law of independent assortment, and principle of dominance. And towards the end of the lecture, we also saw more examples about incomplete dominance, and which are part of non-Mendelian genetics. So, non-Mendelian genetics have all these three categories, such as incomplete dominance, codominance, as well as multiple alleles. So, in incomplete dominance, we saw about the example of the snapdragon flower, where the dominant red flower and the recessive white flower give rise to a pink color flower, which is incompletely dominant. And in the example of multiple alleles and codominance, we saw about the example of the ABO blood groups, wherein the antigens A and B, when crossed together, gives rise to the antigen type AB. And after that, we saw more about the chromosomal basis of inheritance in week 5, wherein we saw experiments performed by a scientist named Morgan involving the fruit fly, and where we saw that the genes for traits reside on common chromosomes, where therefore all the genes are not present on all the chromosomes. And we also witnessed some common definitions like wild type and mutant, and what do they mean. And then we also went on to discuss about certain sex linked genes, and how father can transmit the sex linked genes only to the daughter, and mothers can pass the sex linked genes to both sons and daughters. And after that, we saw more about the definition of recombinance and recombination process, and how chromosomal aberrations lead to genes. And we also saw more about the karyotyping, and we also saw about the karyotyping is the process where we get to know the information from all the chromosomes. And then we also saw chromosomal aberrations and how they lead to diseases such as Down syndrome, Edwards syndrome, Pateau syndrome, and Turner syndrome. And then towards the end of the lectures, we also saw more about the classical genetic experiments, where we discussed more about the Griffith experiment, the Hershey and Chase experiment, and the Michelson and Starr experiment. The Griffith experiment was an experiment which was made to perform to prove that something is being passed on from generation to generation as genetic material. And in Hershey and Chase experiment, the doubt between DNA and protein being the genetic material was cleared, as DNA was proved to be the genetic material. And in Messelson and Stahl experiment, it was proved how the DNA replication happens, whether it happens in a conservative model, semi-conservative model, or in a mixed model. And through Messelson and Stahl experiment, we came to know that the DNA replicates by a semi-conservative model. And towards the end of the week 5 lectures, we also saw more about the bacteria and viruses, where we saw the different shapes and sizes of bacteria. How do they eat, and how do they move around using chemotaxis, and how do they divide using binary fission. And we also saw how most bacteria have circular DNA, which can be used for recombination and inducing new mutations. And then we saw more about the gram staining process, where bacteria can be divided into gram positive and gram negative bacteria, which is based on the presence of peptidoglycan on their cell membrane. And then we saw more about the gram staining process, where we saw about crystal violet staining, gram staining, and then saffron and counter staining. And then we also saw more about bacterial diseases such as anthrax, recycling, and then we also saw more about their functions, which involves decomposition, nitrogen fixation, and symbiosis. After that, we saw more about viruses and their general properties, and saw more about their virus applications as well as their diseases that can be caused by virus. And then we also saw more about the classification of influenza. And in week 6, we also saw more about the cell cycle, cell division, and developmental biology, where we saw where we initially saw the difference between somatic cells and reproductive cells called gametes, and then we also discussed more into cell cycle, how the cell cycle is divided into the G1 phase, S phase, G2 phase, and the M phase. The M phase is where the actual division occurs, whereas the G1, GS, and G2 phase are resting phases, where the cell prepares itself for division. And even in the M phase, there is a mitosis phase and a cytokinesis phase where the cell divides itself. And there are two types of cell divisions, such as mitosis and meiosis, where each type of cell has its own, each type of cell divides itself according to whether it's a somatic cell or a reproductive cell. And after that, we also saw more about the developmental biology process, where where we saw about the fertilization of an egg and a sperm to form a embryo, and how it undergoes further processes such as cleavage, gastrulation, um, organogenesis, and metamorphosis to form the adult once again. And then we also saw more about the animal cloning experiments, and then we also saw about the stem cell biology and the applications of stem cell biology, and how it is used to study the make the karyotyping of the child. And we also saw some of the experiments by which we can do that, mainly amniocentesis and then chorionic fluid extraction. And then we also saw a potential application of the developmental biology, which involves IVF, which is a ex vivo fertilization process. And then following that, in towards the end of week 6, we also saw more about evolution and how Darwin's theory of evolution suggested that um, the different types of animals are subjected to different environmental cues, and depending on that, their features are changed over time. And we also saw more about the Hardy-Weinberg theory, where Hardy-Weinberg principle suggested that under certain conditions, the frequency of homozygous dominant alleles and frequency of homozygous recessive alleles added with the heterozygous alleles will always be the same. And then we also saw some examples of convergent divergent evolutions. And following that, in week 7 week 7 and week 8 almost mostly dealt with amino acids and proteins. And we saw how amino acids are the building blocks of proteins, and how there are several amino acids which lead to different kinds of proteins. We saw how a peptide bond is formed between two amino acids, and how different addition of several peptide bonds result in the formation of a protein. And then we saw more about the stereoisomerism of a protein. And then we also saw about the different folding dynamics of amino acids, which range from at the primary structure, the secondary structure, which can be divided into either alpha helix or a beta sheet, the tertiary structure of a protein, and then a quaternary structure. And in the subsequent lectures of the week 7, we also saw more about proteins and proteomics. We saw the ladder of how anything can be studied through genomics, transcriptomics, proteomics, metabolomics, and then phenomics. And then we saw more about the phenomena of alternative splicing, where a single genome can be alternatively spliced using a spliceosome, which means that the coding regions can be altered by a spliceosome to produce multiple proteins from a single gene. And then we also saw how different kinds of protein uh, analysis can be made. This is from both week 7 and week 8. So in week 7 we saw more about techniques such as our two-dimensional electrophoresis, where one dimension is through the segregation by isoelectric point, whereas the other separation is through your molecular charge to molecular weight. And then we saw more about other techniques such as your mass spectrometry, where the where mass spectrometry is used to study the mass of a certain peptide of our interest. We also saw a lot of we also saw how a mass spectrometer is divided into ionization technique as well as your mass analyzer. We also saw a lot of analyzer ionization techniques such as your soft ionization techniques such as electrospray ionization and MALDI, and we saw other mass analyzers such as quadrupole or time of flight, and we also saw other advanced techniques such as orbit wrap, fusion orbit wrap, or QTOF, which involves two mass analyzers which are lined next to each other. And following that, we also saw other other techniques to study proteins in proteome, which involves your functional which involves high throughput sequences like microarrays. Microarrays are divided into two types. One is the abundance based microarrays, and one are the functional based microarrays. And following that, we also saw more about label free detection techniques of proteins, which involves the in which we mainly discussed about the surface plasmon resonance. In surface plasmon resonance, we discussed how the SPR works and how the analyte ligand binding results in the change in the intensity of the SPR plasmon intensity with respect to the incident light, and how that can be used to study the binding kinetics between the analyte and the ligand of your interest. And towards the last part of the lecture, we saw also got two tutorials which are based on bioinformatics tools. We saw a lot of bioinformatics tools which can be used in different ways to get different information. Most of them include NCBI, nucleotide blast, cluster omega to see multiple sequence alignment, PyMol to study docking of proteins and to visualize them, and protein data bank to extract details about proteins, string DB to study the interactions between different proteins. This is very important in the context of interactomics. Panther DB, we also saw more about Panther DB and PatchDoc, which give a quantitative information about your protein interactions. And then towards the last part of the lecture, we saw more about your uh, ethics and research and publication. We discussed about reasons for scientific paper retraction. The course instructor started with the fact that a lot of scientific manuscripts are being retracted in recent times, and we also saw how those scientific paper retractions are being happening. And we saw some examples of image manipulation where the image is being manipulated to form desirable data. We also saw other types of research misconduct such as your fabrication of data, your falsification of data, as well as your plagiarism. And then we saw how scientific misconduct can be done using unethical practices and using fraudulent data. And then we discussed more towards the causes of research misconduct and how can we as researchers improve ourselves to reduce this scientific misconduct and do proper science. So that's all the recap I have for the whole um, course. So time now is 3:25. So maybe I'll leave that time till 3:30. So between that, if you have any doubts, you can proceed to ask. So if you have any doubts, feel free to put it in the chat box. Okay, if there are no doubts, we'll get started with the problem sessions. So, before starting, I have so the problem statements I have designed it to have some theoretical problems as well as some uh, mathematical problems which you have to calculate. So you can solve them one by one. So some of these questions are very basic questions. So I'll just give you some time to go over the questions. So first question is, what is biomimicry in engineering? So there are four options. This is a multiple choice question. So the investigation of micro creatures inside natural environments, the imitation of natural designs and processes in engineering, the use of artificial intelligence in engineering, the development of new biomaterials using bioengineering. So this question is pretty simple. So these are the kind of questions you might get in your exams. So biomimicry is nothing but, so there are some natural phenomena happening, and those natural phenomena have some features which can be exploited if we engineer the same kind of phenomena for other things. So it's the imitation of natural designs and processes in engineering. So I'll go to the next question. Which of the following statements is or are incorrect about viruses? So option 1, viruses do not contain a ribosome. Viruses can make protein. Viruses stay latent outside host cells. It is considered as a connecting link between living and non-living organisms. So the question is, there are four options are given about viruses, and you have to find the incorrect statement. So if you go by option by one, viruses do not contain ribosome. They do contain a ribosome. Uh, Viruses can make protein. This is not always true because viruses can make protein only within a host cell. Viruses stay latent outside host cells, which is correct. It is considered as a connecting link between living and non-living organisms, which is also correct because it is somewhere in between. It can't live outside the host cell. So the correct option is viruses can make protein, which is incorrect. So I'll go to the next question. Plastids and mitochondria are two important organelles of a plant cell. Which of the following features are exhibited jointly by both of them? So, as you can see, this is a multiple select question. So, there are four options given. So, out of these four options, you can choose multiple options which you feel correct. So, the question is, plastids and mitochondria are two important organelles of a plant cell. Which of the following features are exhibited jointly by both of them? And the options are having a double-stranded DNA, having 70S ribosomes, having 80S ribosomes, and having prokaryotic origin. So, if we were to discuss the correct answer, so we know that uh, this is not the common feature. 80S ribosomes are not the common feature between plastids and mitochondria. And but both plastids and mitochondria, we know that they are from a prokaryotic origin. They have a 70S ribosome and they have double-stranded DNA. So the correct options are the first three options: having a double-stranded DNA, having 70S ribosomes, and having prokaryotic origin. So I'll go on to the fourth question. Look at the image provided carefully. What is the function of the plasma membrane specializations shown in image? So I can look at the image again. So I'll repeat the question. Look at the image provided carefully. What is the function of the plasma membrane specializations known in the image? So this kind of protrusions are what they are asking between plasma membrane. So these are like protrusions which are used to attach themselves to each other. So the correct answer is to provide attachment to each other cells during severe stress. So Ramesh is attending a biology. Fifth question is Ramesh is attending a biology class. His teacher asked to observe onion cells under the microscope. He observed a cell with two separate nuclei and a thin cell plate dividing the two chambers and leading to the formation of two cells. What stage of mitosis did Ramesh observe under the microscope? So I'll repeat the question once again. So we already went through the cell cycle and development uh, part. So in that, Ramesh is attending the biology class. His teacher asked to observe onion cells under the microscope. He observed a cell with two separate nuclei and a thin cell plate dividing the two chambers and leading to the formation of two cells. So there are two separate nuclei and a thin cell plate dividing the two chambers and leading to the formation of two cells. So the cells are like almost like this. So I can draw it and show it to you. So they are saying that, let's just say this is the microscope area. I'm just drawing it blankly. So he is observing two kinds of two cells with two nuclei, and there is a thin cell plate dividing both of this. So they are asking what stage of mitosis did Ramesh observe under the microscope? So when the cells are almost about to divide, this exhibit the process of cytokinesis. Cytokinesis is the final stage of division where the cell starts bifurcating to divide the both the nuclei and the cytoplasm between to form two cells. So if you have no doubts, I'll go on to the next question. So which of the following characteristics distinguishes eukaryotes from prokaryotes? Which of the following characteristics distinguishes eukaryotes from prokaryotes? So the answer options here are improved cell motility due to protein-based flagella, streamlined translation, presence of cytoskeleton to provide rigid structure, and compartmentalization of cellular processes to increase the efficiency of processes. And the obvious answer here is the fourth option. Eukaryotes are better able to compartmentalize themselves in comparison to prokaryotes to increase the efficiency of processes. So I'll go to the next question. Over time, the proportion of Indian women who are at risk for cervical cancer has increased. Towards an effort to raise awareness through scientific research, IIT Bombay has recruited a cohort of 100 women to be screened for cervical cancer risk factors. Which is the strategy would you like to follow? So the options here are protein microarray, multi-TOF, PCR, and both multi-TOF and PCR. So the key here for solving this question is the sample size, which is 100 women. So all of them have to be screened for cervical cancer. So what will you do? So once you see the sample size, your immediate option for solving this question should be protein microarrays. So the correct answer will be you will screen the serum samples using protein microarray for autoantibodies. Because protein microarrays will have a lot of uh, for performing a protein microarray you need less sample size, and you can use the same sample from the same patients to screen for multiple risk factors in a microarray, and you can use a large amount of samples for performing the protein microarray in comparison to your multi-TOF or PCR, which not only will take time but will also be very very less efficient when you are using more number of samples. So I'll go to the next question. So this is a simple theoretical question. A molecular technique in which DNA sequences between two oligonucleotide primers can be amplified is known as polymerase chain reaction or PCR. So we have already covered the concept of PCR multiple times in week 2 and week 3 of lectures, and there you can see that we use two oligonucleotide primers to amplify the sequence of interest. And we also saw how primer design can be efficiently done to study the effect of changes of nucleotides in primers as well as to study the effect of those changes on diseases. So, ninth question. In SDS page, the protein sample is first treated with detergent sodium dodecyl sulfate SDS to. So they are asking what is the purpose of the detergent sodium dodecyl sulfate, which is SDS. So the option is one, making protein negatively charged. Second one is make the protein positively charged, renature the protein, and maintain the pH of the protein in its pI, which is your uh, isoelectric point. And the answer is pretty theoretical. It is to make the protein negatively charged. So sodium dodecyl sulfate is surfactant which covers the protein of your interest, and it gives all the proteins a uniform negative charge, so that when you run the gel across the electrical gradient, instead of charge to mass ratio, the proteins will only be separated based on the mass, since they have a uniform negative charge, and since they have a uniform negative charge, they will be attracted to the anode in a similar way. So the option is option 1, make the protein negatively charged. And for the last theory question, Pranjal is a junior researcher at a cell culture lab, and he requires a specialized cell line for one of his experiments. Fortunately, many frozen vials of the identical cell line stock are in the possession of his senior. However, the senior still won't give him the access to the cell vials despite repeated requests. Following many failed efforts, Pranjal was given one cell stock. Nevertheless, he was curious, so he conducted a STR, which stands for short tandem repeat profile, to verify the legitimacy of that specific cell line, and discovered that the senior had given him the incorrect cells. What action plan does he have? So Here, the situation is simple. It's a theory based question. It's also an ethics based question. So, what can he do? If he has received a cell line from a senior and he has found out that it's an incorrect cell line. So, what should he do? Like, they are given several options: tweeting, speaking to the supervisor, lab meeting, as well as to relocate the lab. The correct answer for this is simple theoretical. So he should discuss with the supervisor first and seek assistance using him. So that the supervisor will talk to the senior. If the senior had accidentally given the wrong cell line to him, then it will not be a research ethics issue. But if the senior had purposefully given the wrong cell line to him, it will be a serious research ethics issue. So the correct answer would be he should speak with the supervisor first and seek assistance suspecting a research ethics issue. So that's all I have for the theory questions. So after this, all of them are problems. So till now, if you have any doubts in the theory part, feel free to ask. I'll give you time till like 3. Now time is 3:47. I'll give you time till 3:55. So some of the sample discussions of the theory part questions are discussed now. So we can move on to discuss the mathematical problems which might come in your exam. So we will get started right away. So question number one, a team of researchers has purified two novel proteins A and B which have lengths of 300 and 400 amino acids respectively. And to study the intracellular localization of the proteins, they were expressed in a cancer cell line fused with two fluorescent packed proteins GFP and RFP having molecular weights of 27 and 25 kilodaltons. What are the approximate molecular weights of the fusion proteins? Given the average molecular weight of an amino acid and water are 110 daltons. So this, I think we have already discussed this problem in your assignments as well. But these are some of the sample problems you might get in your uh, actual exam. So this is a problem which you might need to solve using your calculators. And the key here is they are given the protein length of the protein, each protein A and protein B, and they are also given the molecular weight of GFP and RFP. But the difference is they are given the average molecular weight in Dalton, but they are given the molecular weight of GFP and RFP in kilodalton. So here 1 kilodalton is 1000 daltons. So you have to make the calculation according to that. So we just have to calculate for, let's just say A. So all of them are different values. So if you calculate for one, you will automatically get the other protein's value. So for A, I'll just write it here. It will be 300 amino acids. If you guys want to solve it, I can also give you like 5 minutes for now. Then I can proceed to solve it. You can try to solve it and then you can let me know what the answer you're getting. Then we can try to solve all together. I can give you 5-10 minutes to solve. So the simple way to go around and solve it is multiply the length of the amino acid with the average molecular weight of the amino acid and then add the molecular weight of your GFP or your RFP depending on what the protein is conjugated to which protein. So, option A. So, okay. So, I think that is the correct answer, anyways. But we'll anyways try to solve the answer. So, yeah. So, as I said, you have to take the length of the individual amino acid, multiply it by your molecular weight. So, for protein A is 300 amino acids long, and if you multiply it by the molecular weight of amino acid in water, which is like 110 Dalton, and then if you add it with your molecular weight, so it is 27 kilodalton. So it will be 27,000 Dalton, because the answer is also in Dalton. All the answers are in Daltons. It's safe for us to calculate this in Daltons. So if you do it in calculator, you will get around 60,000 Daltons. So since A is 60,000 Daltons, B also will automatically be 69,000 Daltons. So your correct option will be option A. If you do the same thing with B, you will get 400 into 110, which will be 44,000, plus your 25,000 will give you 69,000. So we'll move on to the next question. So using size exclusion chromatography, you have, once again I'll just my user pointer. Using size exclusion chromatography, you have purified a 100 kilodalton protein. To check the quality of the purified protein, when you performed a page, which is polyacrylamide gel electrophoresis, in the presence of SDS and beta mercaptadinol, it yielded two bands, each having a molecular weight of 20 kilodalton and 30 kilodalton, respectively. However, a native page gives you a 100 kilodalton band. So this is the SDS page and this is the native page. In SDS page, you use SDS and beta mercaptadinol, and in the native page you don't use SDS and beta mercaptadinol. So below is the representation of both the gels. What could be the best explanation of this scenario? So they are given a lot of options. So one option is protein has three subunits. Protein is a homotetramer. SDS page was faulty. Protein is a homodimer. So for solving this question, you need to know what the function of SDS and beta mercaptadinol is. Can anyone say what the function of SDS and beta mercaptadinol is? Like it is already, we already discussed this in the lectures as well as in the weekly recap. Sure. I'll show you the previous one questions as well. But we will finish the. So there are like four more questions. So after this, I can show you. And always after this, I will also be uploading the questions and the slides as well as the recording to the Google Drive. So whenever you feel free, you can go to the Google Drive and then you can access it. So SDS we saw in the previous questions that it is used to impart a negative charge to all the proteins, thereby imparting a uniform negative charge so that all the proteins travel uniformly towards the anode, and the beta mercaptadinol is used to cleave the disulfide bonds between your proteins. Thereby, if you have dimer, trimer, or tetramer, it is used to break those bonds, break those disulfide bonds. So we can go over all the four options one by one. So the first option is the protein has three subunits, one having 100 kDa, one having 30 kDa, and the last one having 20 kDa molecular weight. So if that is the case, then the SDS page should have had a band in the 100 kilodalton range, and the native page should also have two bands at 30 kilodalton and 20 kilodalton range. So this option will not be holding true. The second option, the protein is either a homotetramer of 20 kilodalton or a homotetramer of 30 kilodalton subunits. So if it's either or, then only one band should be present here, and in the native page also this band won't be present. So this also is not wrong. This also is wrong. Or if it's a homotetramer of 20 kilodalton, then the molecular weight here will be 80 kilodalton because it's 4 into 20, which is 80 kilodalton. Whereas if it is a homotetramer of 30 kilodalton units, the total molecular weight will be 30 into 4, which is 120. But we don't see a band in the native page at 120 or at 80. So this option also doesn't hold true. The SDS page was faulty, hence has multiple wrong bands. So in this option also not correct, because even with SDS it's forming the true triplicate, and even in the native page it's forming true triplicate. So the SDS page cannot be faulty. The last option is the protein is a homodimer of two heterodimers, 20 kilodalton and 30 kilodalton. So here they are saying that protein is a homodimer, and each part of the dimer has a heterodimer of 20 kilodalton and 30 kilodalton. So if we do that, the total mass will be 20 plus 30 plus 20 plus 30, which gives you 100 kilodalton. So if you run the native page, it will be 100. But if you use beta mercaptophenol in the SDS page, all the fragments will be cut into 20 and 30 kilodalton bands. So you will be getting 20 bands in the 20 kilodalton and the 30 kilodalton range. So the correct answer will be the protein is a homodimer of two heterodimers, 20 kilodalton and 30 kilodalton. Is this clear? Is this answer clear? So I'll go on to the next question then. So Rashmi was initially growing bacteria in 15N media, and then she moved them to the 14N media. Let us assume that DNA replication follows conservative model. Then which of the following bands would appear after density gradient centrifugation after two generations in 14N medium? So we know that there are several models proposed for uh, replication of DNA, which is semi-conservative, conservative, and a mixed model. So conservative model is what we observed through the Michelson style experiment. But here in this question, they are saying that the replication follows a conservative model. So do people want to go ahead and solve the question? So in a conservative model, I can draw the question. Uh, so so it's given a conservative model. So I'll just give the first cycle for example. So if we have two strands of DNA, so after the first cycle, the parent strands will remain as such, but your daughter strands will have two extra sets of DNA. But in the semi-conservative model, each of the parent strands will have one daughter strand. So this is the difference between your conservative and semi-conservative model, and this was the one which was proved through your Michelson and Starr experiment, whereas this is the one which was proved through the which is being asked in the question. So if you do this, if you do this same replication for one more time, what will be the uh, what will be the density gradient centrifugation? You can take your time and solve the question and let me know the answer. So option three, one band of light DNA and one band of DNA with heavy density. Yeah, that is the correct answer. So basically, what will happen is, so if you follow this for, so if you follow this for a second cycle, you will get the parent strands will still be intact, but the imagine if the parent strand, this is the daughter strand. So the parent strand will generate two daughter strands, and the daughter strand will generate additional two daughter strands. So we have one entirely heavy chain, and we have three entirely light chains. So if we run DNA's centrifugation on density gradient centrifugation on that, we will be getting one band of light DNA and one band of DNA with heavy density. So the answer is one band of light DNA and one band of DNA with heavy density. Option 3. So 14th question. In a population of 1000 individuals, a particular gene has two alleles, capital A and small a. The allele frequency for capital A is 0.6 and for A it is 0.4. According to the Hardy-Weinberg equilibrium, what will be the expected number of heterozygous individuals in this population? So you have to use the Hardy-Weinberg equilibrium, and they have given you A and capital A and small a frequencies. So using that, you have to find out what will be the expected number of heterozygous individuals in this population. So at this point, uh, must be familiar with the Hardy-Weinberg principle, which is the p square plus 2pq plus q square is equal to 1. So basically, in this case, you have to find out what 2pq is. Can you people solve it and let me know what the answer is? Yes, 480 is the correct answer. So I'll just display the answer here. So this is how you go on to solve the question for this Hardy-Weinberg equilibrium. So I told you that the Hardy-Weinberg principle states that p square plus q square plus 2pq is equal to 1, where p represents the homozygous dominant frequency, q represents the homozygous recessive frequency, and 2pq represents the heterozygous dominant frequency. So according to the question, they are given that the P, which is dominant allele, is 0.6 and Q is 0.4. So you just substitute all these values into the equation and use a calculator as well. So you have to find 2pq, which is the heterozygous dominant frequency. But using this frequency, you also have to find the expected number of individuals. So for first, you have to find the frequency. So the frequency 2pq will be 1 minus sum of p square plus q square, which is equal to 0.48. After doing the calculation, you will find out that is 0.48. So if the frequency is 0.48 in a population of 1000, then the number of individuals will be frequency into population, which is 1000 into 0.48, which is 480. So the correct answer will be 480 for this one. So last question, same. You have to use the same Hardy-Weinberg equilibrium, but in a different way. The autosomal recessive illness exists in a community that is also thought to be in Hardy-Weinberg equilibrium. The condition affects 16 out of every 100 people. So it is autosomal recessive, so it affects only like 16 out of 100 people. What are the diseases allele frequencies in the population? Also, what should not be done by the individuals in the group to keep the population in Hardy-Weinberg equilibrium? Choose the best combination. So this is also another part of the Hardy-Weinberg equilibrium. So you just have to find out what the so basically they are given the q square here. 16 out of 100 is they are given the q square. So you have to find out p and q from that, and then you have to justify what reason should be given for Hardy-Weinberg equilibrium. So using the calculator, you just have to find the square root and then so use the other part of the Hardy-Weinberg equilibrium principle, which is p plus q is equal to 1 to find the answer. So does anyone want to go ahead and solve this? It's fine. I'll go ahead with the answer. So the calculation is pretty simple. So I already said that the q square is 16 out of every 100. So you just have to find q square, which is 16 divided by 100, which is 0.16. And so you just have to find q, which is the small m here. Yeah, the option is A. So you just have to find qm, which is the root of 0.16, which is 0.4. And for p, you just have to subtract q from 1, which gives you 0.6. So the capital M is 0.6 and small m is 0.4. And so the automatically the other answer also suffices to the this thing. So to avoid being in Hardy river equilibrium, the individual should not migrate to other places. So that's all. Yeah, this is the answer. So I'll just go over the answers from the first once again. Yeah. So I'll just go over the answers once again quickly. So first question is, what is biomimicry in engineering? So the answer is the imitation of natural designs and processes in engineering. Option number two. So moving on to the next question. Which of the following statements is there are incorrect about viruses? Viruses do not contain a ribosome. Viruses can make protein. Viruses stay latent outside host cells. And it is considered as a connecting link between living and non-living organisms. Does anyone want to attend this question? It's a very so compared to the last five questions, these are like theoretical questions, theory-based questions. So some of these options might not hold true in all the cases. So those are the incorrect options. So the correct answer here is option B. So the fact that viruses can make a protein is always not correct. So that is the once again. So that is the incorrect answer. That is the incorrect statement, which is the correct answer. Sorry. So I'll go to the third question. Plastids and mitochondria are two important organelles of a plant cell. Which of the following features are exhibited jointly by both of them? So this is a multiple select question MSQ, meaning that out of all the four options, more than one option can also be correct. So the correct answer among this is three options: having a double-stranded DNA, having 70S ribosomes, and having a prokaryotic origin. So these are the uh, common features which are exhibited jointly by plastids and mitochondria. So question number four. Look at the image provided carefully. What is the function of the plasma membrane specializations shown in the image? And these are the protrusions that they're talking about, and we have to see what kind of function do they serve? And the answer is they are here there to provide the attachment to cells to during severe stress. Option number D. Next question. Ramesh is attending a biology class. His teacher asks to observe onion cells under the microscope. He observed a cell with two separate nuclei and a thin cell plate dividing the two chambers and leading to the formation of two cells. What stage of mitosis did Ramesh observe under the microscope? So we can clearly see from the diagram it's cytokinesis. So I was already giving explanation about cytokinesis. So when the cell is almost about to divide, cytokinesis happens. So it just bifurcates and finally it forms into two different cells. I'll move to the next question. So which of the following characteristics distinguishes eukaryotes from prokaryotes? And the main difference here is compartmentalization. So eukaryotes can efficiently compartmentalize compared to prokaryotes to perform different functions so that they can increase the efficiency. So the answer is compartmentalization of cellular processes to increase the efficiency of processes. I'll go on to the next question. Over time, the proportion of Indian women who are at risk for cervical cancer has increased. Towards an effort to raise awareness through scientific research, IIT Bombay has recruited a cohort of 100 women to be screened for cervical cancer risk factors. Which is the strategy would you like to follow? So, once again, the keyword here is 100 women. So, you would like to perform the same thing for a large number of samples, and you don't know how many cervical cancer risk factors are there. So, you would like, if you collect a sample from 100 women, you would like to minimize the sample usage by using it for a lot of sample factors. So, which one of the options will you go for? So, protein microarray, multi-TOF, PCR, or multi-TOF and PCR combined. Does anyone want to attempt this question? So, okay, the answer is option A, protein microarrays. Question number 8 is a simple question. A molecular technique in which DNA sequences between two oligonucleotide primers can be amplified is known as polymerase chain reaction or PCR. So we had already discussed the concept of PCR over week 2 and week 3. So next question. In SDS page, the protein sample is first treated with detergent sodium dodecyl sulfate to do what? And the answer is sodium dodecyl sulfate is a surfactant, so it helps to make the protein uniformly negatively charged. So the answer is option number 1. And the last question is regarding a ethics based question in which guy is given a given a wrong cell line from his senior. So for that, the right option to go with is option number D, which is to talk to the supervisor and to seek assistance from him before suspecting a research ethics issue. So all of these are problem based sums. So that's all I have for the problems for the this thing. So and time now is 4:25. So the session will end at 5. So till that, I'll keep the room open for uh, doubts and other questions. So if you have any other general doubts or technical doubts, you can ask here till 5. Can I know how many questions will be asked for exam? So it depends on, so that is not sure. So it depends on how many marks. So how many marks is fixed for your side, but how much weightage is each question given? So whether it's given zero point 0.5 marks, or so if any given question is given zero point 0.5 marks or 1 mark or 2 mark, it depends and it varies. So only depending on that you will be given the number of questions. But you need not worry. But number of questions will be designed in such a way that you can solve it within the stipulated time. So within 3 hours. So can I skip a question for solving it later and do the one second. So can I skip a question for solving it later and do the rest of the questions, and can I able to come back to that question? Definitely you can. So if the I still believe the NPTEL system is using the TCS Ion software. So you can always leave that question and then solve the other questions and then come back to the question. Or if you are not sure that, and it will not be like you have to solve that question to go to the next question, and it's not like you can't go back and change the answer as well. So there will be multiple options like solve to answer the question. So either you can choose the green, so there will be green buttons, purple buttons, and white buttons. So if you are not sure that whatever you have attempted is not correct, you can mark that question for review, solve the other questions, and then come back. That mark for review you can do even if you haven't attempted the specific question. And by any chance, can I know when the exam is scheduled for you people? I mean this exam. So is there any 23rd? Okay, Sunday. Okay, nice. So I think before the exam starts, I think you people will be getting instructions on uh, how to attempt, and most probably you might also be getting some model question paper to solve before 2 days or 3 days before the exam. You might be getting some model. If they are asking you to attend the exam through a software in the exam center, then it is highly likely that they will ask you to they'll give you a demo of the software and ask you to familiarize with the software through mail. So they will do that. And is there any practice question paper for exam preparation? So I think if you log into your course, there will be a tab called week 0. So most of the week 0 questions, they are almost like for exam preparation only, except that some questions you can't some questions might require you to access some websites like NCBI or Blast or something like that. So those questions you can avoid because I'm not sure how those questions would work. But apart from that, I think the other questions you can use that for your exam preparation. I hope that is clear. Are there any other questions? Previous year question paper, I don't think it's possible because there is a chance that some of the questions from previous year are also given to this year as well. So even for us TAs, we can't see the previous year question papers. So if no one has any other doubts, so I thank you all sincerely for all the engaging questions that you have asked over the past few weeks, past 8 weeks. It has been a engaging journey for me as well, learning several concepts and revising some concepts along the way. So I thank you once again all, and I wish all the best for your exams. So I'll upload all these clips to YouTube, and I'll upload all the slides to the Google Drive link as well. So you can access all the recordings as well as the Google Drive lectures whenever you want. And feel free to ask any doubts through YouTube or through my mail ID, which is given at the uh, beginning of every slides that I have given. And once again, I'll thank you and I wish all the best for your exams. Thank you.